Good morning, everyone. Welcome to Virtual Breakfast today on August 14th, 2025. My name is Teresa Crook. I am a MSU Extension Field Crops Agronomist Educator. I am located at the Saginaw Valley Research and Extension Center, and my position is partially funded by the Soybean Committee. This morning, we have Dr. Marisol Quintanilla, MSU Associate Professor and Extension Entomologist in the Department of Entomology. Her research focus is discovering solutions to plant parasitic nematodes in Michigan field crops, including soybeans, wheat, potatoes, and sugar beets. So with that, I'm going to have Dr. Quintanilla bring up her slides and begin her presentation.
Good morning, everyone. I see there's quite a few people join, more than 100. So glad to see all of you. Today I'm going to be talking about soybean cyst nematode and nematodes in potatoes. I'll also mention a little bit about sugar beet cyst nematode. So let's get started. So first going to start with soybean cyst nematode. To know how soybean cyst nematode looks, I have a couple of pictures. So you can see a soybean cyst nematode right there. It looks like a grain of sand. And this is a nitrogen nodule. Nitrogen nodule is about 10 times bigger. And here's the soybean cyst nematode again, that little white dot right there. Here's a beautiful picture by Angie Teeny, the nematode diagnostician at MSU. By the way, MSU has excellent plant diagnostic lab. You can submit samples, soil and root samples to the MSU plant diagnostic lab. They are identified by Angie Teeny, an excellent diagnostician, excellent nematologist. If it's soybean cyst nematode, it's covered by the checkoff dollars. But you can see there, those white balls right there, those are soybean cyst nematode females sticking out of the root. Here's another picture. You can see the nitrogen nodules and the soybean cyst nematode females that almost look like grains of sand stuck on the roots. And they're about the size of a grain of sand. They're about that size. All right, so for the life cycle, you can see to start with an egg, it hatches, goes inside the root, sets a feeding site, and then the males develop their free living, and they'll breed with they go around free, and they can mate with the females, and you can see the female becomes enlarged, and the plant sets a special feeding site for it. And when she dies as a cyst full of eggs, like a case full of eggs that can persist for years, even decades. Here's another picture of the life cycle, and you can see actually real pictures of the egg, of the juveniles inside the root, the sausage shape when they're becoming enlarged, male and female. There's another male and female, a female with laying eggs well connected to the root, and a cyst, dead female full of eggs. So soybean cyst nematode is the most damaging soybean pathogen in North America. It takes the most yield as a pathogen. It's a serious problem in North America. You can see some symptoms there. And it reproduces quickly. I'm not going to go through all the details. Here you can see a map of the United States with soybean cyst nematode. Angie has added a few counties there. This is not an updated map, but the gist of the story is that soybean cyst nematode is found pretty much everywhere where soybean is grown. In pretty much all the lower half of the lower peninsula, the lower part of Michigan, the Midland area. And wherever soybean is grown, pretty much soybean cyst nematode has arrived. Most soybean cyst nematodes found, in fact, in Michigan, 94%, already can successfully reproduce. And the most common source of resistance, P. 788. We did a trial in which we rotated Peking back to back. Peking is another common source of resistance, the second most common, P. 788 back to back, and then rotated with them. And after this trial, we found that if you rotate Peking back to back, the resistance breaks down quickly. But on the first year, it works really well. In the first year, it works really well, and it increases yield, reduces nematode, but it doesn't last forever. So we recommend that you don't replace P. 788 with Peking, but that you rotate with Peking. You rotate with non-hosts, you like corn and wheat or other things. And when you use different sources of resistance, that you rotate them. We also done trials with chicken manure, and we found with a 10% with 90% confidence, not 95 our standard, that the chicken manure does reduce soybean cyst nematode. So some important management practice for soybean cyst nematode: test your field and know your numbers. Submit the samples to plant diagnostic lab. Rotate resistant varieties and sources of resistance. Rotate with non-hosts such as corn, wheat, and cucumbers or whatever. Consider using a seed treatment nematicide. For sampling, send your samples to the MSU Plant Diagnostic Lab. It has the instructions on how to sample and how to submit. And they're covered by the Michigan checkoff dollars. Maybe Teresa can share the link with you guys on the chat or something, that the Plant Diagnostic Lab link. Okay, so there's a few sources of resistance. The most common, again, is Peking and P. 788. Rotating with non-host is the most important thing you can do. Most cover crops are non-host. I already mentioned the management practices. We did a survey. For soybean cyst nematode, and we found the only thing you need to focus is the last bar right here. That is cyst nematode heterodera. And you can see this is the different counties. We did a survey of the different counties, and we can see the greatest concentration of soybean cyst nematode was found in the Monroe County, and second most frequent is Clinton County. The number 2, meaning that the nematode can reproduce in P. 788, was found in every sample. So confirming previous observations that most Michigan soybean cyst nematodes are able to successfully reproduce on P. 788. You can see, so this is the HG type. So 2 found in every sample. 1 is Peking, and you can see that Peking is found like in half of the samples. So yeah, Peking resistance is happening, but at the less frequency, probably because fewer people use Peking. So we evaluated wheat for soybean cyst nematode control. So we found out that wheat is a non-host. That's already was known. We thought that it might be a trap crop. It isn't a trap crop. We found those results based on contamination, but we did find it's an excellent rotational crop with soybean cyst nematode. Besides being a non-host, it has many other benefits. Yeah. I'm not going to go through all the details there. You can see a picture there with a soybean cyst nematode going in a soybean root. Okay. These are just different experiments with wheat in which we also did experiments with entomopathogenic nematodes to control soybean cyst nematode, and it didn't work. So the egg hatching, we can see pretty much the conclusion. These are different entomopathogenic nematodes. So entomopathogenic nematodes don't have any impact on soybean cyst nematode. There are trials that they do affect negatively root knot nematode, but for some reason not soybean cyst nematode. So that is it. One thing that I want to mention is that we've done several experiments with compost and manures, and we found that compost and manures that are stinky, or you would call them strong, something like chicken manure or swine manure, kill nematodes and decrease hatching of soybean cyst nematode, while high carbon compost that are less strong, you could call it, not as much nitrogen, not as potent, some things like dairy manure or plant-based compost, they induce hatching. They help nematodes hatch. And this is something that you can play with. Maybe if you're going to plant corn, you can use a high carbon compost that will induce them to hatch, which would be a suicide mission, because if they hatch in corn is a non-host, so they would die. But if you're going to plant a susceptible host, you can use a stronger compost, something like chicken manure, which actually kills them. So potato, we've done quite a bit with compost and with root lesion nematode Verticillium dahliae. I'm going to, this graph right here, this is different concentrations of compost. Like, for example, this first one is 95% sand, 5% manure compost. And you can see, and this is a kind of an old experiment that was published. You can see in phytopathology, you can see the link down there by Emily Cole, my student. You can see clearly that the layer ash blend from Morgan composting pretty much eliminated all root lesion nematodes at a 5% concentration. Dairy dew had almost, which is a dairy manure, had almost no impact or in mortality in root lesion nematode. And the poultry manure, also very effective to reduce. And we also did a similar experiment with concentrations that were lower, like 0.1%, 1% concentration. And we found similar results. The poultry manure and the layer ash blend were the most effective in reducing root lesion nematode. And layer ash blend also improves yield. Okay, we don't have so much time. So poultry manure also seemed to be most effective in reducing root lesion nematode in our in other experiments. And you can see here similar results: poultry manure, the poultry manure plus Vited, poultry manure alone, and etc. Had it rated, and it increased the yield. It's the highest yield. That's the graph about yield. Okay, my student wanted to find out if any of the effect of the manure was based because of the microbes or because of the biology. So we did autoclave versus not autoclave. So we autoclaved the manures and we extracted the microbiome. And the moral of the story here, and all experiments in my lab are repeated twice in order to publish. That's a basic requirement for publication, is repeat an experiment twice. So you can see that compared to vellum and untreated, all the composted manures reduce root lesion nematode significantly compared to untreated or the chemical control. But you can see also that the autoclave, poultry manure autoclave, was not, uh, um, did not, in general, they were about similar with the non-autoclave that you can see in both experiments right there. So you can see autoclave versus not autoclave, not significantly decomposed egg, autoclave, non-autoclave versus, yeah, not much different. In we also did experiments in the greenhouse trials in roots, and again we found similar results, that the autoclave versus not autoclave were not much different, but they were significantly different from untreated control. So mainly it's not biological. Most of the effect is chemical, okay? Most of the effect is chemical based on um, things like butyric acid, acetic acid, etc. And the systems that mostly reduce nematodes also increase yield the most. You can see poultry manure autoclave, which in general was the one that decreased nematode the most, also had the highest yield. That's the light blue line. That's the plant height. It increased plant growth. Tuber number, same thing. The poultry manure also was the highest amount of potatoes, tuber growth. All right, so you can see again, the winners was the poultry manure. I think I'm just gonna end. So I'm going to thank my lab team for all their work. And I don't know if there's any questions at the end, but I'm done. So thank you so much for inviting me.
Thanks, Marisol. All righty, so we're going to go ahead. Actually, I'm going to, because I want Marty to be able to speak just briefly, at least as far as his comment, and then we'll go to the questions in the chat. I'll start the chat questions from the top and go down. I know Marisol's answered a lot of them, but not everybody can see them in the chat if they're driving. So, Marty, if you want to go ahead and get started.
Yeah, thanks, Teresa. So just a quick note that southern rust on corn is pretty prevalent this year. So there's a few fields that are going to experience some yield loss because of southern rust. Most rusts overwinter down south and move up every season. So that's what's happened. We've had favorable conditions for that to happen. And these warm conditions that we've had, the 80 degrees Fahrenheit is around about ideal for southern rust. And so that's another reason why it's blowing up. There's a lot of susceptibility amongst the hybrids. There's probably going to be some differences, but overall, I think we generally assume that they're fairly susceptible. So I guess just be out, scout. If you're not sure, send a sample in. It is hard to distinguish common rust from southern rust. There'll be an article up with MSUE News later today to help you distinguish. But again, get a sample in. I can try through photos to distinguish, but it's much better to get a sample in and do some spore measurements just to double check. So I think that's what I have on that one. Thanks, Teresa.
Thanks, Marty. Are there any other specialists that want to chime in for anything?
This is Chris here. Western bean cutworm is really low this year, and it's just not me. I have some University of Wisconsin does a ton of trapping, and I got something from their newsletter that said that the count this year is below the 10-year average for them. And for comparison, last year was the highest Western bean cutworm in their trapping network. They averaged over 200 moths in a trap. So I think that could explain why we have this plummet. A lot of times you get a lot of moths in one year, a big population, and then disease and predators kind of builds up and then they kind of collapse, like, that sort of thing. I have other people at Ag Expo that said they scouted many fields and saw like one egg mass. And my colleague in Ontario, who has graduate students working on this pest, they've only found one egg mass all year. So I'm not saying that they're not there. You could have a highly attractive field in an odd spot. But if you're going to look for something at the end of the season here, with Marty's ear molds, whatever, fall armyworm is sort of creeping north a bit. We don't typically get it. And we got earworm early, sweet corn pest, but sometimes you see it in field corn. Those are just things to kind of think about. But from the Western bean standpoint, it's pretty darn low. And I'll be at the Sevrek field day, which tree is next Tuesday, correct?
Correct.
I guess I better know when the field day is. And if I will have cut my BT corn plot by then, and if there is any insect pressure, I'll probably see some of that and be able to talk about that. Thanks, Tree.
Thanks, Chris. Any other educators or specialists? Alrighty, I'm going to start from the top and go down as far as the questions. There were a few questions about, can nematodes on roots be seen with the hand lens, or can you see them with your naked eye? If you could talk about that, Marisol.
Yeah, the question is kind of general because it just says nematodes, but I think they mean soybean cyst nematode. And for soybean cyst nematode females, the cyst, like midsummer, starting in August and of course all the way to fall, you can clearly see the female cyst in the roots. You can see them with your naked eye. If you have good vision, they just look like little grains of sand. Of course, you can get greater focus and clarity by using just a regular magnifying glass or anything that magnifies. For other nematodes that are not soybean cyst nematode or any kind of cyst nematodes, no, you generally cannot see them. But for soybean cyst nematode, the female cysts are pretty big and they're like grains of sand, and you can see them with the naked eye.
Okay, thank you. Hopefully that clarified some questions that people had about that. Some of the other ones had the next, and I don't know how much detail you want to go into some of these questions, but at what rate of application tons per acre does manure impact soybean cyst nematode hatching?
Okay, so we have done several experiments with different rates for things like chicken manure that pretty much kills nematodes and prevents hatching. The greater the concentration or the higher the rate, the more the effectiveness. But of course, you can't be adding like huge amounts of chicken manure, like there's economic and environmental, limiting factors. So, but most of our experiments in the field are somewhere between 1.25 tons and 2 tons per acre. And most growers that do apply manures or compost, they apply between that amount, between 1 ton and 2 tons per acre. Like I said, in general, we found the greater effectiveness with greater concentrations. Yeah.
Okay. And the other question that was coming up toward the top was about cover crops and which ones would be non-host versus host, if I may paraphrase it a little bit.
Yeah, for soybean cyst nematode, almost all cover crops are non-host. The legume-based cover crops, most of them are poor host. Okay, so because soybean is a legume, then other legumes can be good hosts. But most cover crops, like let's say vetch and alfalfa, whatever, or they're poor hosts. For dry beans are good hosts. I know dry beans are not a cover crop, but they're a crop, So yeah, so any dry beans are excellent hosts for soybean cyst nematode. Most other things are poor crops. Unfortunately, there are some weeds that are that are hosts. So controlling weeds is important.
Okay, there was a question also about an interaction or if there was any impact as far as glyphosate with nematodes.
No, glyphosate has no significant impact on plant parasitic nematode. And most things, most things don't. There's some fungicides that have impact on plant parasitic nematodes because there's some fungicides that act both as a nematicide and a fungicide, but most other pesticides do not, or herbicides do not. All pretty much herbicides that I know do not have an impact on nematodes.
Okay. And then any studies on nematode populations and damage to dry beans? I wonder if they probably asking an
Yeah.
economic threshold.
Yeah, I have not worked with dry beans. Dry beans are susceptible to soybean cyst nematode, like soybean is. So for thresholds, etc., I think we would have to look at other people's work, but the work that done on soybean is very applicable to dry beans because they're similar hosts. Whatever works to control soybean cyst nematode in soybeans would also work in dry beans. Of course, there's no sources of resistance to the best of my knowledge. There's no varieties that are resistant, but rotating with non-hosts, etc. Yeah, but I have not worked with dry beans.
Do we know what attracts or repels nematodes to certain crop root systems?
Yeah, I guess nematodes know what they like. So soybean cyst nematode recognize the root exudates of soybeans. In fact, it helps them hatch. You know, when you have this cyst, the juices, you know, the juices, the smells, and the chemicals that coming out of the roots induce their hatching. There are some nematodes that feed on just about anything, like root lesion nematode, and they'll just enter any root. But nematodes that are very host specific, like soybean cyst nematode that have a narrow host range, they only feed on certain things. They're attracted on the smells or the root exudates of the plants that they feed on.
Okay. Are there any effect from manures and compost on verticillium?
Okay, so not much. Okay, not much. Most of our experiments showed greater effectiveness on the nematode than verticillium. We did have significant difference on our chicken manure reduce. Um, um, boy, I forgot the name. Um, when you have verticillium in potatoes, the potato inside will have a line, like a ring around it. And I know the name very well, but it escaped my head right now. But so it did have a significant impact on that. So it does have an impact, but we couldn't find when we did like vert numbers in the soil or in the stem, but in the potato, yes. So I think there is an impact, but it's been that impact has been hard to quantify for us, and it's not as much as in nematodes.
Okay, a couple more, and then I think there's at least one question for Dr. Andreessen. What is the basis for trying EPNs for soybean cyst nematode management?
Okay, the answer of that one is because there's been research showing that entomopathogenic nematodes significantly reduce root knot nematode that helps to control root knot nematode. There are papers on this, and we thought maybe they will have a similar impact on soybean cyst nematode. And the paper indicated that it's likely because of the chemicals in the EPN. The entomopathogenic nematodes carry a bacteria, symbiotic bacteria, and that symbiotic bacteria produces certain chemicals that negatively affect plant parasitic nematodes, in this case, root knot nematode. And we thought, you know, maybe this is working with soybean cyst nematode, and that chemical can be identified and then used as an effective nematicide. But it turns out it doesn't work on soybean cyst nematode like it does root knot nematode.
Okay, then how does one separate chemical and biological effects?
Well, for our compost, we attempted to do that by autoclaving the compost, therefore killing the living, organisms. And with most of our results, and this experiment indicated to us that most of the impact of the manures is based on the chemicals, or in the manures and not on the not directly biological.
Okay, I think I captured all those for you, Marisol. There's one question about weather. Jeff, any prediction on the first frost for the southern Lower Peninsula?
Well, we can. Whether it's any good or not is another story. The reality of that one, it's really tough more than maybe a week to 10 days out to have any skill. That's just the way that that works. So the statistics are very, very important here and relevant, and they've been changing. The date of this first freeze of the fall with time has gotten later in the year than it was in past decades. But and I do promise. So for Southern Lower Michigan, just to give you an idea, most areas that would be at the end of the first week of October or in the second week of October is when that would occur. That would be the first 32. We also need to look at, certainly for some crops like corn, we'd be better off using a lower threshold like 28, the first hard freeze. That would be a week and a half to 2 weeks later on average than that. So that would be well into the later part of October. That's the median, the 50th percentile, and we typically see all of those dates occur, most of them within 2.5 to 3 weeks either side of that. What I will do, and the graphics for this are currently unavailable because of some computer maintenance right now, but I promise I will bring those next week and talk about the looking ahead here. But I guess the bottom line is, again, it's a really tough game to do very far out, and I will look at that certainly, and if there's any threat of an early coming, we would be looking at that medium range forecast guidance, and we would be looking for, well, northerly, northwesterly flow aloft, bringing Canadian air into the region. That's when it's going to happen. But typically, Mother Nature doesn't give us as much lead time or warning as we'd like. So usually it's within a week or week and a half. So, but I promise to bring more on that next week.
All righty. If there is nothing else, I just want to remind everyone, just like Steve mentioned, that recordings for the virtual breakfast are available on Spotify or Apple Podcasts. You can follow it on Facebook. If there's nothing else, without further ado, have a safe and timely season. Thank you for attending this morning and have a safe harvest. Take care.